The last thing I wanted to talk about when we're talking about microbial growth is how do we grow viruses? And so there's three main ways you would grow viruses in laboratory. So we've talked about growing bacteria on petri dishes and with different media and all different kinds of conditions. And so viruses have their own special things that we have to keep in mind. Remember, viruses need a host cell. So there's three main ways to do this, really generally. One is in the host organism itself. I actually worked in a lab for a couple of years at the University of Florida where we studied viruses that killed caterpillars for potentially to use in agriculture to prevent crops from being destroyed by caterpillars. So we studied viruses that grew in caterpillars, and we actually had caterpillars in the lab. And sometimes we'd expose we need more virus, so we'd expose the caterpillars to virus, and then we'd blended them up in a blender and isolated the virus. It was pretty gross, but we were able to grow a lot of virus that way. If you're talking about animal viruses, you might have lab animals that you grow viruses in. They might be monkeys, they might be cats, they might be things like that. And so ideally, you can imagine some of the things associated with grinding up caterpillars in the lab or having to inject animals and sacrificing them to study viruses and all. And it's not a pleasant thing. It's not ideal. It has its complications. Maybe the mouse already has a viral infection that could influence what virus you're growing or whatever. So we can grow viruses in host organisms. It's not always the preferred method, but it is definitely possible. This picture right here is something that I've done a lot, and a lot of viral research has been done in this way. And what these are is these are agar plates, just like your Petri plates that you would grow bacteria on. And the cloudy stuff in the background is bacteria. And so this one, and then you can see there's these dots on there. So all this stuff in the background in between the dots is bacteria. It's a solid, we call it a solid lawn of bacteria. And there's more dots here, but the stuff in between the dots is the bacteria. What they say clear plaques, what these plaques are is they were created, they are spaces, they're clear spots where there's no bacteria, and they were created by bacteriophage. Remember, bacteriophage are bacteria, are viruses that infect bacteria. So on this plate, a bacteriophage landed right there, got mixed with the bacterial cell right there. And then we let it incubate. And the virus multiplied and multiplied. And remember, every time that virus multiplies and pops out of the cell, it kills the host cell. So that little clear area that they're calling a plaque is a clear zone where there's no bacteria because the virus popped out of it and killed it. And probably killed millions of bacterial cells in that spot. And so for every virus particle that landed here, we can see a clear plaque or a clear zone. If I took that and I analyzed what's inside of there, there's going to be a lot of virus right there. Okay, there's only bacteria in this area, but a lot of virus in these clear plaques. And what you see in this picture are, this one just has a lot higher concentration of virus, and this one has an even higher concentration of virus. So bacterial cell is a host organism. So here we go. Number one way we can grow viruses in laboratory, we can use the host organism, but it has to be the organism that matches the virus. You can imagine that if you're studying human viruses, not a lot of humans are going to volunteer to be the host organism for your research project. So we have to think of better ways to be able to grow viruses than just the host organism. And a very common and very effective way to grow viruses in laboratory is doing something called cell cultures. So in this diagram, it's just showing you that if you want to grow, you want to study a virus that grows in the lungs. Maybe you want to study SARS-CoV-2. It grows in lung tissue, but nobody's going to volunteer for your study to let you grow virus in their lungs. So what you do is you go in and you get a little biopsy of human tissue, and there you have some cells. And then there's all sorts of lab techniques where you take those cells and you clean them and you separate them and get rid of any other any other tissues that are there, and you just have these cells. And then what we can do is we put them in a petri dish or a flat dish with some kind of medium, some kind of food, and we just grow the cells. So now we've got lung cells, and we can infect these lung cells with the virus to study a human virus that infects lungs. So we call this a cell culture. So creating a cell culture, we would use some host cells and grow them separate from the host in the laboratory. And we could use that to grow viruses. Here's a picture. This is a continuation of the cell culture. This is kind of how they end up looking in the laboratory. Here's a little plastic container that's sterile, and it's got some sterile media in it. And you grow those cells that you isolated in this medium, and they just kind of fall to the bottom. And you can actually look at them under the microscope. Like I could take this little flask and I can put it look under my microscope and look right through there and see the cells without even opening it. So this might be what you see under the microscope. Here's some human cells that are kind of settled into this liquid down here. And so you could inoculate this with a virus and grow virus and study what does the virus do to the cells. So two ways that we grow viruses in the laboratory. One, we can use the host animal, host organism. Two, we could use cell cultures. And a third common way to grow viruses in a laboratory is using fertilized chicken eggs. Now, and this works for some viruses. It doesn't work for all viruses, but some viruses, what you do is you get chicken eggs, and you have, it's fertilized, so it's got an embryo growing in it. There's the yolk sac. And different viruses work in different ways, but you can inject the virus into that egg. So you've got these actively growing host cells inside of this egg. And for some reason, I guess I don't know, for some reason, a lot of different viruses, even human viruses, can grow on chicken eggs. And so what they're showing you right here is here's somebody that's injecting these eggs with some virus. And they're going to grow this virus up. Here's another more industrial looking one where they're injecting eggs with virus. And then what we end up doing is after that virus has grown, and it takes, I don't know, it takes a few weeks, and it will end up killing the embryo because the virus is going to multiply and kill that embryo. And then in the laboratory, you're going to crack open all these eggs and purify the virus out of them. Now, the thing that you're probably aware of is that a lot of times our flu vaccines are made this way. We could make flu vaccines, just like, let me just back up. Like, what is a flu vaccine? A flu vaccine is a weakened form of a virus. So we inject the egg with a weak form of the flu virus. It multiplies that. We put it into our bodies, and our bodies think that we've been infected with the flu. Our body doesn't know that it's a weak form of the virus, but the vaccine is actually a weakened virus or a dead virus that we can put in our bodies and get immunity to the flu, to influenza. And here's a little bit of how they do that. Just, a little bigger of a diagram. Here's our fertilized egg. We inject this weakened influenza into it. The virus is going to multiply, and it's going to kill the chick that's developing. And then what you're going to do is there's a whole fancy stuff that goes on that you end up making the vaccine out of that. Now, a lot of what we heard with the pandemic and with making vaccines from eggs in general is, one, it's relatively inexpensive, and it's an effective way to get a lot of vaccine. The trick is it takes a little bit of time, because what do you have to do? First of all, you've got to get millions of these very clean eggs. You don't get them from your regular old farm. These eggs are produced in facilities where the animals are under relatively clean environments. I mean, chickens are dirty anyway, but these are environments that they're try to keep the chickens under as clean as possible. And then the eggs are harvested in very clean ways, and they are also cleaned, and they've got to be tested to make sure they don't have other viruses, they don't have bacteria. These are highly controlled farms where all these eggs come from. In fact, it's I think there's like secrets about even where they're located. So, and it's my understanding that Pennsylvania is a big state for producing these really high quality eggs. Um, but anyway, so you've got to go through all the technical things to get the eggs made, and then you've got to inject them, and then you've got to grow the virus, and then you've got to test them, and then you've got to purify the virus, and then you have to make the vaccine, and it takes more than 6 months from the time. In fact, it takes closer to a year, but from the time that you start making the vaccine to the time you can use it. So when we were talking about the pandemic, when we're needing a vaccine as soon as possible. This is a new virus. It could end up killing a lot of people. We need a vaccine quickly. And it's a new vaccine. Making it in eggs can be time consuming, which is how the RNA vaccines got started. And we'll talk more about that when we get to that chapter. But anyway, 3 major ways you can grow viruses in a laboratory: in the host animal, in cell cultures, and in chicken eggs. And that's the end.